Welcome to our Ginkgo webinar today, where we will highlight our deployment capabilities in fermentation, scale-up, and purification for proteins and enzymes. My name is Mervyn D'Souza, and I'm part of the business development team at Ginkgo. I have over 25 years of experience in technology and product development, R&D strategy and creation, and innovation. I've spent most of my career leading global teams and driving commercial development and execution at a number of large multinational and smaller companies. The basic principles that drive success don't really change, regardless of where you're at. So many of you are well aware of Ginkgo as an organism engineering company with an end-to-end horizontal synthetic biology platform that can be used across a number of different fields like food, ag, or pharma space. What some of you might know is that for the last 15 years or so, we've been honing our platform and capabilities and acquiring technologies that have expanded both our breadth and our depth scientifically and geographically. The unparalleled power of our code base and foundry teams at Ginkgo working together is the foundation of our ability to deliver unique solutions to our customers. As you can see, our foundry we have access to massive metagenomic sequence libraries. We've got the best-in-class microbial host strains. We're not limited to our own host strains. We can work with customers' host strains as well. And then with our automation and our protein engineering tool called OWL, using AI, along with our NCAPs and screening technologies, we can do both rational as well as classic strain engineering and deliver solutions to our customers that help them move along the path to commercialization. We're essentially a one-stop shop and can provide any or all of our modular services to be having some issues. Okay, I think I'm back. So anyway, my apologies. We're essentially a one-stop shop and can provide any of our modular services when it comes to enzyme or protein production. This complements our customers' internal capabilities and enables the execution of your growth strategies and accelerating your products going to market. Today, we want to let you in on what might be the best-kept secret, which is that we at Ginkgo have an experienced team with extensive expertise in scale-up and deploying upstream processes, downstream purification with the relevant analytics. During the commercialization of a new ingredient or product, be it a protein, enzyme, or a small molecule, there comes a critical point, as we all know, where we need to go from that discovery and validation in the lab into the real world. This is a really important process that often determines the success or failure of a new product launch. The ability to effectively, efficiently, and economically scale production to commercial levels can make or break it. This is the focus of our webinar today, as you'll see from the agenda. I am really delighted to have with me today 3 of our best from our Ginkgo Foundry team, with a total of 60 years of process development and scale-up experience between them. My new philosophy for 2024 is don't just tell me, show me. With that in mind, Kevin Hettenbach will tell us about our current Ginkgo deployment and scale-up capabilities. Derek Samuelson and Chris Leonetti will then show us how our Ginkgo team creatively deployed both upstream and downstream processing expertise to support the scale-up of enzymes and proteins for our customers. Without any further ado, I'm going to hand it over to Kevin to kick us off.
Hi, Mervyn. Thank you, Mervyn, for a nice introduction. Hi, everyone. My name is Kevin Hettenbach. I'm a principal project engineer in the deployment group as part of the foundry at Ginkgo Bioworks. I have a PhD in chemical engineering and a master's in environmental engineering and a professional engineer's license. I worked at Ginkgo for 8 years and support technology transfer of our customer products from laboratory to commercial scale. I manage our customer commercial manufacturing operations at Fermic in Mexico City. And prior to joining Ginkgo, I worked at Julong Limited for 5 years and Pfizer for 15 years in chemical engineering research and development. Next slide, please. This slide provides background on Ginkgo end-to-end services. In supporting Ginkgo's mission to make biology easy to engineer, Ginkgo is the technical bridge from R&D to customer commercialization. Discovery, development, and scale up to the client's desired endpoint can be challenging. Ginkgo's experience and network is available to enable these activities. The deployment team, as part of the Foundry at Ginkgo, has upstream processing, downstream processing, analytical development and quality control, and pilot plant functions, and an extensive CDMO network to support end-to-end. Ginkgo has proven capabilities to deliver projects to the desired scale and target product profile, from bench scale fermenters at amber 250 milliliter scale to commercial at 50 cubic meters and higher volume. Ginkgo's end-to-end services cover the full range from discovery to scale-up optimization and customer commercialization. Next slide, please. Ginkgo has an extensive CDMO network and in-house pilot capabilities, which provide several advantages to our customers in supporting tech transfer and scale-up. These include avoiding wait times to access CDMOs, which results in reduced time to market, increased tech transfer efficiency, as the team developing at bench scale is the same team tech transferring to a project to scale, continuity and process understanding, which results in reduced manufacturing deviations and delays. Ginkgo has a strong procurement team, which complements CDMO activities, such as providing logistical support for raw material ordering, packaging, and equipment rental. And Ginkgo's CDMO network in US and international consists of over 100 CDMOs, including universities. When vetting and identifying a suitable CDMO, Ginkgo aligns with the customer on scale-up requirements, including production volume, cost of goods, regulatory and process definition, and target metrics. Ginkgo has an established relationship with Fermic, with direct access to pilot with 1 cubic meter and 5 cubic meter fermenters and DSP, and commercial scale manufacturing with a 50 cubic meter fermenter and DSP that supports kosher and food grade manufacturing. The photos on the left show equipment from our West Sacramento pilot plant, including a 30 liter, 300 liter, and 3,000 liter fermenters, and supporting DSP equipment, including a centrifugation, filtration, and spray drying. As part of our first tech transfer from microbial fermentation to our pilot facility early last year, we performed a 3,000 liter fermentation and generated greater than 5 kilograms of material to support successful customer product testing. The West Sacramento pilot plant also supports agricultural based products. Next slide, Mervyn. Ginkgo supports projects across multiple business sectors, including pharma, food, biochemical, and agricultural, and organism types, including bacterial, fungal, and mammalian. In developing projects in these these areas, Ginkgo has demonstrated greater than 10x increase in fermentation productivity, greater than 50% improvement in DSP yield, and up to greater than 90% cost of goods reduction. Ginkgo has a proven track record for developing and tech transferring commercially viable fermentation and DSP processes, with 6 products successfully tech transferred to commercial scale to date. Ginkgo has extensive development and scale experience in upstream processing, downstream processing, analytical development, material generation at lab and pilot scale, process modeling, and techno-economic assessment. The graph on the right illustrates the titer profiles for yeast fermentation, producing a flavoring compound at amber 250 milliliter, pilot 250 liter, and commercial 50,000 liter scales. Equivalent productivity was obtained in scaling up from amber scale to commercial scale. These results demonstrate that the amber fermenters are an effective tool for predicting scale-up performance. Ginkgo has demonstrated equivalent fermentation productivity on several scale-up projects. Next slide, Mervyn. Ginkgo also has extensive development capabilities at our laboratories in Boston. Upstream processing capabilities include fermenter capacity ranging from 250 milliliters to 30 liters. The 10 and 30 liter fermenters are used to demonstrate scalability from optimized parameters determined in amber 250 runs and generate material in the tens to 100s grams range for our customers. We have several examples of generating materials in different business sectors, including pharma, food, and such. Downstream processing capabilities include established unit operations for physical, chemical, and thermal separations. Altogether, over 12 operations, at least we support. For physical separations, Ginkgo has expertise in centrifugation and filtration for optimized biomass separation and protein purification. For intracellular product recovery, Ginkgo has in-depth experience in cell lysis methods, including bead milling and homogenization. Looking at chemical separations, Ginkgo has developed chromatography and ion exchange methods to optimize protein and enzyme purification. And we've successfully used extraction with oils or solvents to obtain a high yield product separation. And for thermal separations, Ginkgo has optimized white film evaporation for high boiling point compounds, such as flavoring products, using low pressure conditions. And we've also successfully used spray drying to meet product specifications for biomass and protein-based products. We also have analytical capabilities, including a wide range of instrumentation to support analyte quantitation and microbial testing. Ginkgo has extensive experience in fermentation and purification development and scale-up of proteins and enzymes, including both intracellular and extracellular express products. Next up, Chris Leonetti will describe a case study in the proteins and enzymes space.
Thanks, Kevin. Hey, everybody. I'm Chris Leonetti. I'm the head of downstream processing here at Ginkgo Bioworks. And at Ginkgo, we define downstream process engineering as everything from the harvest of fermentation broth all the way through purification up until final formulation. I joined Ginkgo in 2016 at a Northeastern University with a chemical engineering degree. And since I've been here, I've seen Ginkgo grow just exponentially from like 70 people all the way over 1,000. And just in that time, I've been able to work on so many different products and processes. It's been really exciting. And I'm happy to share a couple of those here today with you, specifically this next one, and Derek will share a couple others in a little bit. So my case study is going to be on an intracellular protein. And Mervin, if you can go to the next slide. The first thing I want to say is, like, part of why I find this job so exciting is, right, we're able to focus on technically challenging problems, but also economically challenging problems, right? When you see an animal-derived protein at the top of this page, you're thinking about something that is probably trying to displace a well-established product in the animal industry. And, you know, that has like cheap costs, very well supply lines built out, really good cost of goods, and well-established like quality attributes and products, right? Like, so now the customer is coming to us and trying to displace that animal-derived protein with a very simple, like the same protein, just microbe, in this case, Pichia pastoris, derived. And so there's going to be skepticism from the market. There's going to be, you know, boot up costs trying to get this whole process in place as a business model for the customer. And they need to, you know, woo investors, right? They have this great idea and they have limited runway and time to get it done. So what the customer is coming to Ginkgo for is really to accelerate that development time and being able to parallelize all of that regulatory and market work, while also having development work done on a fermentation and a downstream process to make something overall that they can show to a investor, or even just, you know, shore up their business model, you know, profitable, make it more profitable. So in this particular case, when the customer did come to us, they didn't even have a lab build out or even a development team. They just had the idea, the really good product to strain, and you know, that's really where we have a lot of fun stepping in, that's where we go. So Ginkgo's a perfect customer here, or a perfect vendor for this customer and partner, because we had already shown very good proven track record in taking our scaled down model fermentations and bringing them all the way up through 50,000 liter fermentations. We're able to validate at an intermediate scale, develop, make material, you know, first drop or all the way up to a commercial process for our customers, and really not handhold, but partner along the way to get there. In addition, we have the downstream and purification side of things. We have experts in that as well, where, you know, Ginkgo is the organism engineering company. We want to make biology easy to engineer. My job, Derek's job, Kevin's job is to, you know, not focus on the biology so much, but also focus on making sure that the process runs well so that you can actually purify the product coming out of the biology, and in that way make biology easier to engineer. And, you know, so I think we all know, like, this is a case study. It's we're going to put our successes in here, and it's funded by Ginkgo. So the next slide is going to show the outcomes, but it also shows kind of the journey as well. And that's what we want to focus on, because that's really where the fun details come into play. So Mervin, if you can go to the next slide. Thank you. This slide focuses on a two-pronged approach. Ginkgo, strain engineering company, right? We did do some strain engineering on this product, but in the short timeline that we're focusing on here in this case study, it was really about fermentation process improvements and downstream process improvements as well. So we needed to develop like a tech transfer process for both fermentation and downstream for the customer, as well as some additional material for application testing. And we started on the fermentation side of things. So we brought their fermentation process into our scale down model, and we were quickly able to test different process parameters across you know, multiple reactors all at once, really accelerate that development time, and quickly able to get from 5 gram per liter up to 10 gram per liter in the fermentation. So quickly you're doubling your total product output. Great from a COGS perspective, great from a just generating material perspective, right? And awesome to see. Then we take that scale down model, scale it back up to around 30 liters, 10 to 30 liters, and validate it there. This is great both from proving that we can scale from the 250 milliliter reactors, but also now I get more material personally, which is then combining like 250 milliliter reactors together. I can pull from a 30-liter batch and start working on my downstream material more effectively. Still can do it at the 250 milliliter scale, but I like to start with more material. I also find the 10 to 30 liters is more of a sweet spot than, say, scaling to 100 liters. There's really not much difference between the 30 and 100 liters for fermentation, but there's a whole heck of a lot difference in terms of me processing 100 liters of broth compared to 30 liters of broth. Quite a lot more goes into that. So we get that validation at a good pilot scale, right? And now what we want to do from there is make this that tech transfer ready process for fermentation. So we do further robustness testing around the design space of that fermentation to ensure that it can go to any fermentation or fermenter kind of around the world, right? Our customer at the time didn't have a scale-up partner in mind, right? That's like number 47 on their list of things they have to do to get to market. So we need to design a process that could go to a chosen CRO down the line. And different fermenters around the world are going to have different tankage around them, different piping design, different agitations, agitators and agitation rates. So we need to make something that can scale anywhere effectively. So we did that on the fermentation side. And then on the downstream side, the process that we got was very much R&D. Very much focused on just generating that small amount of material at the front end to prove that you have the same product as the animal-derived product. And, the first thing we took a look at was that lysis step. The process that we had was focused on detergent lysis, which, as a lot of people know, if you're getting, detergent lysis is not really going to scale. Too much cost involved there. And frankly, it was about less than 50% yield on the lysis step. And that's the first step in your purification process. You're already losing half your material. That's not going to fly. What we did instead was put this into a bead mill, as Kevin described a little bit before. And that's basically just a big blender for microbes. You put tiny beads inside of it, and everything just crunches in between those beads and pops open the cells. So now we have much better access to that product in the lysate, and we can move on to further steps to actually purify that protein. The purification steps, the more downstream ones for this bench scale process, we're focused on a chromatography-based purification, which is great just for getting that first product again, but not great for scale, especially for, as we talked about before, an animal-derived protein that has very low cost traditionally to produce. We want to move off of chromatography, which is way too expensive for this, and we want to move on to something that's more, filtration based. So we're lucky with this protein that it was in an area in molecular weight size that is more, that wasn't similar to all the native proteins that Pichia produces. So we can actually separate this based on filtration mainly. And that's what we did. We instituted a series of filtrations and cross-flow filtration to generate product that, while it wasn't like 95% pure, it was pure enough based on the acceptance criteria and met the quality attributes of the animal-derived protein. So really important to think with the end goal in mind here. You don't necessarily need something that's 99% pure in this industry. As long as you're matching what you have or slightly better than the current existing product, you can kind of work in there and displace it and save on costs. Overall, we increased the yield just in that 3 to 6 months from less than 40% to over 75%. Huge win. And the customer didn't have to do any of this development, right? They're able to focus on that market entry. They can figure out their regulatory pathway, whatever they need to do with the FDA to get this microbe derived protein into whatever process they're trying to put it in. Throughout this whole thing, we're also able to take that material that we generated to validate the processes and deliver those tech transfer ready packages and give it to the customer so that they can do further application testing. So we were able to generate greater than 10 grams of this sample that they were able to put directly into their formulations. Overall, we estimate that this probably reduced the customer's overall development time and like time to market by about 6 months, which is just a great win, I think, for the customer, but also, further just making biotech and, biology easier to engineer. I think that's a huge win because we have to compete against all these economic factors as well when we're talking biology. On that note, that pretty much wraps up my case study. So I want to pass this off to Derek. It's been a pleasure to talk to everybody, and we'll have Q&A later. Thanks.
Okay, thank you. Yeah, thank you, Chris. Yeah, thank you for that kind introduction. Hello, everyone. It's a pleasure to talk with you this afternoon. Yeah, my name's Derek Samuelson, and I'm a principal process engineer. I have a master's degree in chemical engineering from Cornell University, and I've been working in industrial biotech now for over 20 years. It always sounds funny to me when I say 20 years. Doesn't feel like 20 years, but the math does check out that way. I started my career at a company called Metabolics, where we focused on developing a fermentation and recovery process to commercialize biodegradable polymers from sustainable feedstocks. So I really look fondly on my experience there, and it was a good, hungry startup culture. I then moved to work for a company called Shale-Jardong, or CJ, which is a Korean-based company and one of the largest industrial fermenters in the world. I've been at Ginkgo now for roughly 4 years, and I'm on the deployment team with Kevin Hettenbach and Chris Leonetti, and I lead the upstream process team where we focus on developing and scaling robust fermentation processes. So today, I'm very excited to be with all of you today and to talk about 3 case studies. These 3 case studies using 2 different organisms in which Ginkgo developed strains and processes that quickly, smoothly, and successfully scaled to tens of liters and were able to deliver a substantial amount of quality product, meeting specifications to customers. So the first case study that I'll talk about employs Pichia pastoris, which is a yeast, and we use this yeast to produce an animal-based protein. The second 2 case studies I'll talk about focus on Aspergillus niger, which is a filamentous fungus. And in those case studies, we'll focus on making enzymes. The first case study will focus on the native enzyme, and the second case study will focus on the recombinant enzymes. So with all of that ado, I guess if we jump to the first slide, the first case study I'm excited to tell you about is about using Pichia pastoris to express and produce an interesting animal protein. To do so, we needed to leverage many functions here at Ginkgo, and we do have lots of capabilities, particularly on the upstream side. So first and foremost is our strain engineering capability. We really have some incredible things going on here at Ginkgo. We have a design team that basically, sort of the architects of the strain. A build team is sort of like the construction engineers that make the strains, and then a high throughput testing team that goes through tens of thousands of libraries to sort of identify the best strains. And so we can go through these what we call design, build, test, learn, or DBTL cycles in 2 to 3 months time. So the timeline that we're using is really impressive. So starting from scratch, we can build organisms worth testing in just a few months time. And once this organism is developed, it's then tested in what we call our automated microbioreactors, or AMBER units, in which the strain can be tested and fermentation conditions can be optimized to reach target titers. Now, fermentation is filled with a lot of jargon, so titer might be jargon to some of you on the call. So when I say titer, what I mean is the amount of product per liter of volume of fermentation broth. But there'll be more on the AMBER units in the next slide. And so then once the commercial target titers are reached in these AMBER units, The process is then transferred to our deployment team, which, Kevin Hettenbach and Chris Leonetti and I represent. And for this particular process, we were able to use the three major pillars of Ginkgo's process development capabilities. The first pillar, and what I like to think of as the foremost pillar, is fermentation. I'm a fermentation guy. So we were able to scale the protein production of this particular protein to 30-liter scale in our process laboratory here in Boston. So you'll see that capability here on the picture on the right. The picture shows one of our 30-liter fermenters, stainless steel fermenters, in the foreground. And it's a little bit hard to see, but there's also a 10-liter stainless steel fermenter in the background. In addition, in the same laboratory, we also have other fermenters. So we have 12 by 5-liter glass reactors, 6 by 20-liter stainless steel reactors, and an additional 30-liter stainless steel reactor that is not shown in this picture. And the reason that we have so many reactors is that there's so many projects going on at Ginkgo concurrently. This capacity allows us to work on several projects at the same time. And we use this 30-liter fermenter seen here to produce customer requests when we get customer requests for like protein amounts on the order of somewhere between tens of grams and hundreds of grams of product. We also have larger fermenters in West Sacramento, and I'll talk more about that in a few slides. The second pillar of our process development is in analytics. And really the purpose of our analytics is twofold, right? So we have the high throughput analytics, and this is to help support those design, build, and test teams that I was talking about to help them identify the best strains, right? But second, we also have to have a QA/QC team that focuses on high resolution analytics to assure customers we are delivering products that meet specifications, right? And then the third pillar is downstream processing, or DSP, as it's described in the slides. And it's last here, but certainly not least, right, Chris? We have a stellar downstream processing team. And for this particular case study, I think we utilize some of our core capabilities in recovery to produce this animal protein. So these unit operations are sort of the central unit operations to our recovery team, or like centrifugation, tangential flow filtration, and chromatography using an ACTA unit. In this particular case, like controlling the pH of the product stream through a buffered system was of particular importance because we were trying to preserve the cofactor binding capability of the protein. So this, in combination with finding the right cation exchange resin, helped produce a product that was over and above product specification for purity. Moving on to the next slide. So we're sticking with case study 2 here. This is still with Pichia pastoris, but now we're really going to highlight our strain screening capability. So I think a key feature of Ginkgo's capabilities is the efficiency and speed at which we can develop the strains and processes from scratch that reach commercial targets, right? And I think this Pichia study is a good example of this capability. So again, at Ginkgo, we use these automated bioreactors, or AMBERs, to quickly and efficiently optimize our fermentation processes. And we have an incredible amount of experimental space at our fingertips, right? We have over 200 AMBER reactors at our site in Boston Seaport District. And for this particular project, we ran over 400 AMBER reactors over the course of the time of the study to identify optimal process conditions and feed rates and identify best strains. Right, and so now looking at the graph on the right, there's a lot to unpack here, so I'll try to take it step by step. But in short, I think this graph really shows our strong strain screening capability. So each dot that you see here in this plot represents the titer of the best strain from each design, build, test, learn, or DBTL cycle. So when you're seeing all, that's all AMBER data, but it goes through this DBTL cycle first. And it, you know, goes to the test team. The test team looks at it, goes through literally tens of thousands of strains. You try to get the best hits, and then those best hits are then tested in our amber fermenters. And so the data that you see here represents amber data. So it does take some time to get that first sort of 10x improvement. But I think the key here is that once this DBTL cycle gets liftoff, the improvement is exponential, and we're able to deliver the last like 10x or 100x improvement in titer and reach commercial target in just 1 or 2 DBTL cycles. So I think what these DBTL cycles do really well is really remove those primary bottlenecks, right? And so once those primary bottlenecks to the pathway are removed, the improvement in titer is realized much more quickly. So like soup to nuts, the strain and process took some time to develop, but that was also before we had the learnings that we have now. So if presented with the same test now, we could develop a similar strain expressing a similar protein in much less time, as we wouldn't be starting from scratch. In addition, I'd like to highlight that the cycle time on our design, build, test, learn DBTL cycles has also improved over time, from a 6-month timeline that you see here to now a 3-month timeline. And on the process development side, we can then take the process developed in these amber units and scale it to 30 liter or even 3,000 liter to produce larger quantities in 3 months' time. And I think like when we combine all this together, I think it tells a powerful story that, you know, with the strain screening, the DBTL capability, the optimization capability that we have on the upstream process team, and then you combine that with the downstream team, we're able to develop processes that scale and deliver products at specified quality. We truly have an end-to-end capability. So I think in this particular case, the upstream team used picky-pass stores to express the protein at 30 liter scale, and we produced on the order of hundreds of grams of product, right? And then a fraction of that 30 liter broth was handed off to the downstream team to produce on the order of 10 grams of product. And that downstream team used the unit operations that they're very familiar with: centrifugation, tangential flow filtration, while controlling the pH of that product stream throughout, so that the cation exchange chromatography could maintain cofactor binding of the protein, and we're able to produce a product at a purity far exceeding customer expectations. So I think this is a really good example of how Ginkgo can execute producing a complex protein while using a thoughtful and elegant downstream process. So yeah, moving on to the next slide. So we're gonna shift now from Pichia pastoris to a filamentous fungus called Aspergillus niger. So I think Aspergillus niger is really attractive, both because it is generally regarded as safe or grass, and also because it just has a wildly good protein expression system. Right? It naturally secretes large amounts of protein into what we call the supernatant, or sort of the cell-free liquid part of the fermentation broth. And this is attractive both from an upstream standpoint and a downstream standpoint. Like from an upstream standpoint, we're getting high titers, high productivities, high yields. It's very efficient. And from a downstream standpoint, it is an extracellular protein expression. So it avoids things like lysis and makes for a sort of a more elegant downstream process. And yeah, to reiterate, A. niger really does have like a mind-blowingly good native protein expression. And Ginkgo is working to utilize that protein expression system to make recombinant enzymes. And there'll be more about those recombinant enzymes in a couple of slides. All that being said, I think scaling A. niger doesn't come without challenges, right? So like one challenge that we've had to overcome is that A. niger can be a high viscosity strain. Another challenge is that there are proteases. The protease activity needs to be mitigated so that the protein or the enzyme of interest is stable. And a third challenge is that Aspergillus niger is prone to interesting morphologies and can be difficult to transform, and it's a spore former. For all these reasons, A. niger or Aspergillus niger does not lend itself quite as well to high throughput screening in quite the same way that Pichia does. But at Ginkgo, through our strain engineering capabilities here, I think we have a solution for all of these challenges. For the viscosity challenge, we chose a strain with improved morphology, right? Which makes the strain inherently lower in viscosity than the native wild type strain. And in process development, we changed both the media and the feed strategies to control that amount of biomass. The viscosity is mostly an expression of the amount of biomass. So we were able to control the viscosity of the broth that way. And in this manner, we were able to avoid the mass transfer challenges we otherwise would have encountered had we not chosen a low viscosity strain. And through genetic engineering, we've also been able to eliminate or mitigate both the number and the activity of the proteases. And even though our high throughput screening team does not, or HTS does not work quite as well with Aspergillus niger as it does with Pichia, we can really lean on the experimental space and the number of amber units that we have to do the strain screening through the ambers, as opposed to doing the screening through HTS. All of this has given Ginkgo a powerful tool. We have a chassis strain with both low viscosity and low protease activity so that the enzyme product we produce is stable. And by chassis strain, I mean a good sort of parent strain that you can retain all the good properties that you can pass on to the children, if you will. And so with this chassis strain, we have another organism in addition to Pichia that we can use to produce complex proteins. I want to direct your attention to the top right picture. So this is a picture of our facility in West Sacramento, California. At our West Sacramento facility, we have a 300-liter fermenter and a 3,000-liter aerobic fermenter. So Ginkgo has the in-house capability to scale organisms. It could be Pichia, it could be niger, it could be other organisms, directly to 3,000-liter without having to use a third party. So in fact, like many times we have successfully scaled our fermentation processes developed in Boston directly to the 3,000-liter in West Sacramento. If I could just find a direct flight to Sacramento, I'd be happy, but no, but I digress. But it's really a great validation step that the process developed at the 30-liter scale also works at the 3,000-liter scale. So in Mervyn, if you now click on the video in the bottom left, you will see our 5-liter glass fermenters at work. And these 5-liter glass fermenters are actually from our process laboratory, not in Boston, but in Zeist, the Netherlands. And the Netherlands is a great place to visit, but the team there is even better. Been working closely with that team, and I'm very proud and honored to work with them. And much of the work, or really all of the work that you see with this Aspergillus niger, is really due to that, due to all the hard work of that team. And so in this video, on the top you'll see Ginkgo's low viscosity strain. And on the bottom, you see the native wild type strain. So the difference in viscosity is like apparent to the naked eye, and the low viscosity you see in the top fermenter makes mass transfer much more scalable. So that's it for the introduction to Aspergillus niger. I would now excited to tell you about case study 3. And this is where we're focusing on overexpressing the native enzyme. Now, the native enzyme is a commodity enzyme that we wanted to use as an example to develop a good solid chassis strain or a base parent strain with those attractive qualities we talked about, low in viscosity, low in protease activity, to develop a robust and scalable process. This robust and scalable process could then be used to produce the recombinant enzymes we'll talk about in the next slide. The picture you see on the top right is really repeating the theme that you saw in the video. In the left-hand corner, you'll see the low viscosity strain, And in the right-hand corner, you'll see the more viscous wild-type strain. And you don't need to be a scientist or an engineer to see the difference. It's apparent to the naked eye. And because of this lower viscosity, the process is easier to scale from a mass transfer standpoint. And it's with the scaling in mind that Ginkgo chose this low viscosity chassis. Now, the plot on the bottom right shows the degree of process improvement with the Aspergillus niger strain. So we've been able to improve the strain both through engineering. We overexpressed the native enzyme, but also through a lot of rigorous process development on the fermentation side. And when I say process development, I mean specific things like media optimization, process condition optimization, like optimizing the dissolved oxygen, the temperature, the pH of the fermenter, and also feed strategies to control things like the biomass that's developed. In doing all this, we were able to increase the overall production at the 30-liter scale by 4 times. Pretty impressive numbers. And the productivity of the native enzyme is so impressive that we're able to produce kilograms of enzyme in our 30-liter fermenter, that same 30-liter fermenter that was pictured a couple of slides ago. In addition, we used the unit operations, familiar to you now from the previous case study, centrifugation, tangential flow filtration, and chromatography to recover and purify this enzyme of interest. And finally, moving on to case study 4. So this is, again, continuing with the Aspergillus niger story, but this really brings the story home for Aspergillus niger. This is really about taking that chassis strain I've been telling you about and now using that chassis strain, in this case now, lessening the native expression and focusing on expressing the recombinant enzymes. So I think this really highlights the power of a good working chassis strain. It really bears a lot of fruit. So now, like using the same concepts and the same workflows we use with a native enzyme, we've been able to improve this enzyme production between 1.5 and 14 times for 4 distinct recombinant enzymes. And again, this is with A. niger. A lot of the development and improvement has come from fermentation process development. So optimizing those same things that I've been talking about, dissolved oxygen, pH, temperature, media formulations, and feed strategies. And in addition, we're able to piggyback on all the learnings from scaling that we learned with the native strain when we scaled the recombinant strains. And these recombinant strains were also scaled to 30-liter and reached or nearly reached commercial targets. So in closing, I'd like to say, I think with these 3 case studies that we've shown, I think we've shown that Ginkgo has 2 organisms in Pichia and Aspergillus niger that can be used to produce targeted proteins and targeted enzymes for your process and your business, right? We can do the same thing that we've done with these case studies for the products that you want to make. And we have the know-how, the capabilities, and the capital equipment to develop the process, to scale the process, and to deliver high-quality products that meet specifications. I hope that you'll consider using Ginkgo's end-to-end capabilities. It's been a real pleasure presenting to you today, and I'll now turn the webinar back over to our moderator, Mervyn D'Souza. Mervyn, take it away.
Okay. So thanks, Kevin, Chris, and Derek for not just telling us about what Ginkgo's deployment capabilities are, but actually showing us how Ginkgo provides customer solutions to the case studies that you shared with us. We have had the audience paying attention and have a few questions for our panelists, and we'll try and get to as many as we can. So the first one I'll direct to Kevin. So this is a question from one of our audience members. Can you provide, we talk about end-to-end quite a bit, during the presentation and in our PR at Ginkgo. Can you provide an example of a successful Ginkgo end-to-end project, Kevin?
Yes, thanks, Mervyn. Yeah, so joined Ginkgo in 2016, and actually during that time we were developing our first um, product for to scale to commercialization. So Ginkgo's first successful end-to-end project involved strain optimization, fermentation, and DSP development, and tech transfer to pilot and commercial scales for a flavoring compound. Highlights of the development work include a 10x improvement in fermentation titer and a 40% improvement in DSP yield. The process was successfully transferred to commercial scale in 2017, and Ginkgo has been supporting production at Fermic to meet our customer volume requirements since 2017.
Thank you. I think maybe on the flip side of that, we had another audience member who wanted to know if Ginkgo took on projects that were focused mainly on process development and optimization. You know, we're known as the organism engineering company, but I think after today, the secret is out with the three of you and what the deployment team can do. So maybe address that to Derek, and, you know, Chris, welcome to chime in as well.
Yeah, thanks, Marvin, for the question. I think that's a great question. So yeah, Ginkgo is the organism engineering company, right? I mean, I think I even have a t-shirt to prove it. But yeah, we also can do things start to finish, soup to nuts, as I like to say. We're a one-stop shop and can do things end to end, right? And so I think we're also like kind of like to think of ourselves as like a good restaurant. Like I do think we have like a complete menu, right? And so I think, yeah, if you could do things end to end, but you could also, you know, if you just want the strain, we can give you the strain. If you just want the process, we can, if you have your own strain and you just want the process development, we can do that. If you're just interested in scale up, we can also do that. So yeah, I think the strain engineering capabilities here are unparalleled, but we can also do a lot more than that too.
Yeah, and I'll just chime in on that, right? Like, as we talked about earlier, we're here to make biology easier to engineer, and it's not always just the strain engineering and the fermentation to do that. One particular process from during the pandemic, we made an enzyme for was it Aldeveron. So we were just focusing on fermentation process improvement there. So we generated improvements there, and then tech transferred that back into Aldeveron's facility. They were able to take that process and run with it with some troubleshooting help from us. And it, you know, that's also not a traditional fermentation stainless steel reactor. Those are, GMP style reactors too, single use. So fun stuff.
Thanks for chiming in, Chris. And since I've got you on, I'll put you on the spot and ask you another one that came into what type of cost of goods or COGS reductions do you typically see, or can you expect to see when you have an increase in titer or yield, for the process? What can you do with the deployment team?
Yeah, so the easy answer there, right, is that every time you increase the titer, you increase the titer and the yield, right? You're gonna decrease the COGS. Less time in the fermenter, less time doing your DSP cycles. But it really depends on the product. It's more nuanced than it seems. If you're generating a product that needs to go to, like, only really needs 1 or 2 runs a year, that's a lot of CapEx to own your own plant, right? That fermenter is just going to sit idle and you spend a lot of money on it. So you want to combine that with other processes. And it really depends on, as Kevin Hettenbach will can attest to, it really depends on how many times you want to run it, at what scale, and for what purpose. Like, is it pilot or is it commercial at that point? You're going to get varying prices as a result.
Thanks. I think we'll try and squeeze in one more question because there've been a whole bunch that have been coming in. So thank you, audience, for that. So this one is a particularly interesting one, and so what if I don't have internal fermentation and downstream processing capabilities, but I'm interested in leveraging synthetic biology tools to increase efficiency and sustainability benefits for my products? What do I do then? What can Ginkgo do for me? And maybe, Kevin, I'll address that to you.
Thanks, Mervyn. Yes, I think what's been highlighted through these slides and case studies is our end-to-end services and our strong foundation in strain engineering and fermentation and DSP development and scale-up. And Ginkgo has an extensive CDMO network with proven success in tech transfer and scale-up support to pilot and commercial for our customers. And also some of the nice photos shown in these slides show our West Sacramento pilot facility with our in-house capabilities for fermentation and downstream processing to support customer scale-up feasibility and also material generation.
Thanks, Kevin. And that was, I'm glad I had you on the panel. And I know from working with you, you've been scouring the globe, looking and vetting different CDMO, facilities to make sure that while we work with our customers and while you're helping that process, in case customers don't have those scale-up capabilities, we can hook them up with options for them to take that forward. So thanks for that. I want to close out quickly with a huge shout out to our panelists and our audience today. Thanks for your engagement, your questions, the interaction. Hopefully the secret is out. At Ginkgo, when we say we have an end-to-end platform, we mean it, and we won't leave you hanging. If we didn't get to all your questions, we will respond, offline, and you can email us at deploymentservices@ginkgobioworks.com. Please reach out, as I think you'll see that Kevin and Derek and Chris are super excited to work with you, helping you address issues and get you on your way. Be well and have a great rest of the week. And thank you for your time today.
Thanks, Mervyn. Thanks, everybody else.
Mervyn.
Bye-bye. Thanks, everyone.
Thank you, Mervyn. Thank you,
Thank you,
everyone.
everyone.